We're glad you're here for today's EMSL Learn webinar, where our presenter, Sarah Lichty, will be presenting on the Molecular Observation Network and upcoming proposal call opportunities. We do have another webinar that will be happening next month on November 15th on terraforms, which are tunable. I always have to remember the acronym, but custom-made synthetic habitats for researching soil processes. We invite you to register now to attend and to check out more information on the EMSL website. This is a good time to sign up for our email communications and to follow us on social media so that you don't miss opportunities to hear about proposal calls, webinars, and other new opportunities. So we invite you to subscribe. Our links will be posted in the chat. So today, Sarah Lichty, a project manager here at EMSL, will walk you through the MONet program, upcoming proposal call opportunities, and much more. We invite you to stay around until the end of the webinar when we'll have a Q&A opportunity. So we encourage you to post your questions in the Q&A section or the chat. We'll save those for the end and have Sarah answer your questions. We're glad you're here. And with that, I'm going to toss it to Sarah.
Thank you, Genoa. All right, and thank you again for joining us for today's webinar on the Molecular Observation Network. As Genoa said, my name is Sarah Lichty. I'm the project manager for MONet. During this hour, I'll be covering a few important topics, including an overview of MONet and our goals, how our first proposal call went this year, and our plans for future calls. You'll notice that I'm the only speaker today, and that's because we are still awaiting more guidance from the Department of Energy on permitting and compliance. As soon as we have more information, it will be made available on the website and via email. To start, let's zoom out a bit and talk about EMSL and open science. EMSL is a Department of Energy Office of Science user facility sponsored by the Biological and Environmental Research Program. And what that means is that we collaborate on projects with researchers from academia, other government labs, and industry. Open science takes that a step further by engaging with the scientific community to collectively answer large scientific questions by expanding access to cutting-edge capabilities and democratizing access to data, and also tools to understand how to utilize that data. MONet is an open science network which provides, at no cost to you, over 17 advanced molecular and microscale soil parameters, training materials for you and your lab to use when analyzing multi-omic and multimodal data, and provides access to standardized soils data from around the country. You might be wondering, how do I get involved with MONet? Over the next few slides, I'll walk through the process of how you can get from proposal submission to data delivery. This process is what we employed for our first MONet call, which was called the soil function call. As for all other user projects at EMSL, we require a proposal submission through our user portal, Nexus, to collaborate with us. MONet requires a short proposal detailing the sites you plan to sample, as well as a description of how the data from MONet will help you answer your scientific questions. We are currently updating our soil function request form using feedback from our first call, and that new version will be posted along with updated call language sometime in early November. The next step would be to collect the cores. So once a proposal is accepted, a kickoff call will be scheduled to determine when sampling will occur, and a sampling kit, which you can see below, with a standardized protocol is shipped to the PI. While in the field, PIs collect the cores, as well as take a few field measurements, including infiltration rate and soil probe measurements. The PI then records that site information and field data in EMSL's sample metadata file, which will help with data findability and accessibility in our MONet database. The power of MONet is ensuring that the cores and metadata collected can be compared to other cores sampled around the country. So once those cores arrive at EMSL, MONet lab staff process them following standardized protocols. And on this slide, we're highlighting a few of the data types which are unique in terms of their availability to the scientific community. We provide high-resolution molecular data on soil organic matter using FTICR and LCMS. We also have partnered with the Joint Genome Institute to provide metagenomic sequencing, assembly, and annotation. X-ray computed tomography is another advanced data type that provides 3D internal imaging of soil architecture and roots, which you can see in this image. We don't only collect advanced data types, though. We also have more commonplace data, such as texture and pH, to create a complete picture of soil biogeochemistry. And I mentioned earlier that we use standardized protocols to process each data type, and you can explore each of those methods in depth by referencing our protocols published on protocols.io, which we'll put in the chat. Lastly, all data produced will be made available through EMSL's data portal so researchers and modelers can utilize that data. And when we say it's a fair database, that means the data is findable, accessible, interoperable, and reusable. And you'll notice from this slide that we are collaborating with a number of institutions such as JGI, NMDC, and NEON to leverage their resources in our ultimate goal of open science and fair data. This slide nicely summarizes how MONet interacts with the scientific community to connect researchers and modelers with soils data. Here on the left, you can see that researchers are the ones proposing and owning the science by submitting proposals and collecting samples. EMSL then performs the analyses and provides training materials on how to explore and utilize each data type. And other researchers and modelers can access that data and make new discoveries about soil functioning. So now let's recap how our first MONet call went and what we learned from it. Our first call opened on February 14th, 2023, because we love soil, and our internal target was 200 sample sets. That was largely based on our processing capacity as we brought on new staff and set up the lab. We were pleasantly surprised to have a strong response from the scientific community with 28 proposal submissions over the course of 4 months. Since we operate on a first come, first served basis, we had to close the call near the end of June to avoid having to turn away submitters once we'd reached capacity. After closing the call, the MONet team has been hard at work processing the cores coming in on those accepted proposals. Here we have a breakdown of where our first MONet projects proposed to sample. You can see that these sites were distributed across the US, including Alaska and Hawaii. The blue markers represent where MONet PIs proposed, whereas the green markers are NEON sites. We chose to include NEON sites to build off data captured within MONet's pilot project, the 1000 Soils project. And if you'd like to learn more information about that pilot project, we'll include a link in the chat. This graph shows a bit more about what ecoregions the sites belong to. And as a reminder, we're using ecoregions that NEON has defined, which represent distinct regions of vegetation and landforms to be able to capture the diversity of soils across the continental US. You can see we had 15 out of 20 ecoregions represented in this first round of proposals. And I want to highlight that there was a slightly higher percentage from the Southeast, the Southern Rockies, and tundra. And what's exciting about this is that those ecoregions are likely to experience higher impacts from climate change. While we did not evaluate soil function proposals on scientific merit, it is clear that the community is concerned about climate change, and the MONet database will reflect that by having standardized data from all ecoregions, but especially those sensitive ones. We also had 11 out of 12 soil orders represented, so all but spotosols. And again, we were excited to see a healthy representation from these highlighted groups. And without getting too much into the weeds of podology, these orders are relatively high fertility and high organic matter content, and are also at especially high risk from the effects of climate change. You'll also notice that we had a range of land uses from croplands to forested and to grasslands, and a higher number of sites from existing LTERs, kind of leveraging those long-term ecological research plots. Now, one of the aspects that I get most excited about within MONet is the opportunity for graduate students to be proposal PIs and get that experience leading a project. In this pie chart, you can see that we had a proportion of the graduate student participants in this first call, and actually a few served as PI as well. And another portion falling into that early career category, which can be defined as within 10 years of the last awarded degree. We also had a large number of proposals from academia, with a smaller fraction from government agencies and industry, and also a large proportion of new EMSL users, which we are very excited about. So what did we learn from that first soil function call? We saw that we needed to accommodate for higher demand. Our solution to that is to have quarterly calls over the next year to balance the year-round demand without exceeding our processing capacity. We also received feedback from users that our sample set limits were too small to allow for replication. And so I'll show in a couple slides how we've tried to sort of have a solution to that as well. So first off, our tentative schedule is quarterly calls, and we are now finalizing that call language within the next few weeks. So we're aiming for a November 6th call open for our spring 2024 sampling period, which is from March to May. So you can see that we have these quarters so that we make sure we're not closing the call too soon and not allowing for sampling in later portions of the year. And you'll also notice that the submission periods aren't very long, but the proposal process is quick and easy. And so since MONet is first come, first serve, it will be in a user's best interest to submit early in that submission period to increase the chance of proposal acceptance. And again, we will have processing capacity limits where we might need to close the call early, but we will do our best to provide advance notice of when that close date is, now that we know we have a better idea of our processing capacity. Now to the info I'm sure everyone is curious about, which is how many sample sets does my project qualify for? And first of all, I'll define what a sample set is. So one sample set consists of two 30-centimeter long cores and four 10-centimeter long cores. One of the two longer cores will be deconstructed immediately for soil biogeochemical properties, while the other core will be analyzed for soil physical properties. And the four surface cores will be homogenized into the top of the biogeochemical core to account for surface soil heterogeneity. Now, on the next slide, I'll walk through the core processing workflow more in depth. But a change from our first soil function call is that we've increased the sample set limits from 6 to 9 in our small scope project, and from 21 to 24 sample sets in our large scope. And in order to apply for that large scope, we do require documentation of large project status and also sites with complementary open data resources would be ideal. And so the stricter qualifying criteria is to ensure that proposals applying for that higher number of sample sets have sort of that added value of those other data sets to connect to MONet's database. And we encourage potential PIs to email monet@pnnl.gov with questions on eligibility. And as I mentioned before, each sample set consists of 6 cores, but the data associated can be consolidated into a single core. So those purple arrows you can see are associated with our biotic core, which captures the biogeochemical properties of the soil. And we need to process that as quickly as possible since we know the biology of soil can change once it's removed from the field setting. And I want to point out that our current workflow is set up to analyze the top and bottom 10 centimeter sections of the core. Now the orange arrows are associated with our abiotic core, which captures structural properties, including X-ray computed tomography and hydraulic properties, which require an undisturbed core. So you might also be wondering, how do I get my data from MONet or view other data? Like all other EMSL user projects, raw and processed data can be downloaded via the Nexus user portal for PIs with accepted proposals. And data users can access this data on the EMSL Projects and Data webpage, which a link will be provided in the chat. So to improve the searchability of MONet data, a data portal is under development and is scheduled to be released in April of 2024. And then improved functionality will be incorporated into that platform around October of 2024. So hopefully this webinar has sort of piqued your interest about all things MONet. So I'd like to encourage everyone to attend MONet's first community science meeting, which will be 2 days of immersive lectures, tutorials, and hands-on training on November 7th and 8th. And registration is still open for remote attendance. We'll put that link in the chat as well. So we have plenty of time for questions, and I'll pass things off to Linda.
Okay, so I see one question, and I would invite the rest of our attendees to drop all of your questions in the chat or the Q&A section. We can grab those and hand them to Sarah. The first question is from Cameron Richardson. Would a 4-year study site be considered for the large scale study?
That's a great question. And yes, so we do allow for sampling over time, and that is encouraged since we know temporal measurements are really important and valuable. And so that would be something within that request form. We'll have a section that asks how many sample sets you'd be collecting within each year and within kind of what months as well, so we know when to kind of allocate those and expect those samples. And what I would say is the caveat is that we want the first sample sets to be collected during that submission or that first sampling period. So in looking at that call schedule we had when the proposal submissions were being accepted and then when the sampling period was, if you're putting a submission during that time, we'd like at least some of the cores to be taken during that first sampling period, but they can happen in later periods and also different years as well.
Okay, great. Thank you. Question from Deidre. Is there a possibility of sampling over time? Would that involve resubmitting a proposal in future years? Would resubmissions for repeated time points be looked at favorably?
Yeah, again, this is a little bit different, I think, from our last call where we wanted to have maybe resubmissions to be able to capture all of the new cores that we would be processing, but we're working on our side to make sure that we have a tracking system in place, that we know when those temporal samples would be taken in the future, and work to incorporate that into our processing capacity. So only one submission, which will save PIs that headache of having to resubmit every time they want to include more samples. So I'd say just one submission, and we'll have a spot to really detail exactly when you want to take each of your sample sets, so we can do a better job on our side of knowing when those cores might be coming.
Great. A question from Willie. Do you have collaborations with African countries? I live in Africa, in Cameroon. I would like to know if I submit my proposal, if I can be among the selected.
We would love to be able to open it up to other countries, but right now we are only within the US. Part of that is logistically, it will be a bit harder to incorporate and work with other countries, but that is a goal of ours in the future. But this next year, we're focusing just on areas in the US. But EMSL does have different proposal calls, which do involve researchers from all over the world. So I would encourage you to look at other ways to become involved with EMSL. And hopefully in the future, MONet will provide that opportunity as well.
Question from Carl. Can a researcher who submitted a proposal in 2023 submit a different proposal for 2024?
Yes. Yep. So we don't have, I would say largely our guidelines are one active project per PI, but I do think you can submit, and if you have any questions, you can email the MONet inbox to double check. But I think for now, we are still opening up to PIs who want to have multiple projects, and we can sort of discuss that with you and see if the scope is a lot different. Then I think that would be a clear justification for another project.
Okay. This particular question, I'm not sure who it came from, but it says, I am a PI on a large-scale experimental trial that is part of a network of experimental agricultural sites across the state. Would it be best for each site to submit a proposal to stay within the 24 sample maximum? Or is there a possibility of submitting one proposal as a network and go beyond the 24 total samples to accommodate all sites?
Yeah, that's a great question, and one that I think as we're learning more about managing this project, we will likely have calls in the future that do focus on even larger collaborations. We don't have anything set up currently, and I know John Barger is on the call, and so he might want to say a few words extra, but I would say as it stands, I think submitting those separate proposals, but we would be able to see internally that they're linked together. But I'll let maybe John jump in if he has some thoughts.
I didn't know I was going to be unmuted so quickly. I was just trying to type a response. Yeah, I think we've discussed this this exact situation where you have a large research network in the context of a large scope proposal. And the answer is we would certainly like to consider it as a large scope proposal if you think it fits. And in that case, we would encourage you to reach out and let us know that you'd like to do that. We are really interested for these large scope proposals. There are 3 important criteria we're looking for. First of all, we want to get that geographic diversity and we want to build up the database. Number 2, we really are looking to make sure that you have really the ability to make the lift and support the large scale sampling. And that's often the case across a distributed network. Third, we're really looking for ancillary data types that are going to be in the public domain. So that instantly adds value to MONet data as well as your data, right? Of course. So You know, we'd want to know if the data are in the public domain and just make sure that the value that would be created by co-locating spatiotemporal co-sampling between MONet cores and data that you've already accumulated would be able to be available to people. Those are all important considerations. Yeah, reach out and let us know. It's just this doesn't fit neatly within a simple description of a large scope proposal. So rather than trying to add a whole bunch of new specific instructions, we thought it'd be best to just say reach out to us.
Okay, great. Thanks for jumping on, John. Appreciate it.
Sure.
And Sarah and John, if you want to add to this too, and it might kind of fit into what you were just talking about, but Sony has a question about how do you categorize small and large scale studies?
Yep, and this is something that it is tricky. It's hard to really, draw a line and say, some PIs fall within one category or another, and so we realize it's a little bit vague. I would first of all think critically about what John just mentioned of both the capability of your lab group to be able to take the number of cores that a large scope would entail. The sampling equipment is, we send it out, we provide instructions of how to use it, but it does require, your team to go and do that work and be able to coordinate with us and get everything shipped back to us as quickly as possible so we can process it. And so that is one consideration of if you do have a large project that has the capacity. And also, as we had mentioned, we are looking for sort of larger scale projects that are sort of funding your team's time to do those sampling efforts. And so that dollar amount, we're not going to say it has to be more than X number of dollars. But really you can think of those large projects as probably being potentially, $1 million, multi-million-dollar project, large scale, being able to ideally be across eco-regions. So we talked about those NEON eco-regions. That is something that we are trying to get samples from all of them. Even if we have some samples from 15 out of the 20, we want to really be able to capture that heterogeneity in soils. So thinking of that too, of, maybe there is a use case where we have a lot of different types of land uses in a small region, and that might be a justification for doing a large scope project there. But in general, you can think of typically spanning large areas so that we're capturing more with those 24 sample sets, because we don't want to be awarding too many of those projects and then we are able to reach, we aren't able to reach as much of the community and help, more groups with answer their scientific questions. Hopefully that helped a little bit.
Yeah, no, that's good. Okay. Katarzyna, I believe I hope I didn't completely butcher that. The question is, I am at a PUI. Are these proposals limited to R1 institutions?
Nope, they are definitely not limited to R1 institutions. We're even encouraging, say, citizen scientists to apply as well. We want this to be, we don't want there to be barriers to working with MONet. And really, if you have the team to go out and do the sampling, we provide that kit and the standardized protocols, but we really do, it's in that vein of open science that we want to be able to serve the scientific community and not have it only be, research institutions that maybe already have the capability to do some of this research without us. We really do want it to be a collaborative effort.
Great. Hey, Jean Lim is asking, would MONet consider soil cores from seagrass beds?
Oh, interesting. I would say right off the bat, we don't want, John says no, we don't want inundated soils. And part of that is that we can't maintain that, microenvironment once it's taken from the field. We don't have any sort of, way to process it anaerobically. So John's short answer is no. I gave you a little longer answer, but unfortunately, we can't maintain the integrity of that soil for processing.
Hey, Oduwu, his question is, does sample collection allow for cores from replicated plots or only one core per treatment?
I would say part of that reason that we upped the sample set limit from 6 to 9 was exactly for that reason. We understand that replication is important. And when you're looking across treatments and hopefully capturing any sort of treatment differences, having that replication can be really important. So I would say, yes, we do encourage replication. We realize that maybe it doesn't allow for, 4, 5, 6 replicates, but we're, hoping that at least upping it from 6 to 9 will provide for a bit more replication while also ensuring that we can have a lot of different projects accepted through MONet.
A question from Kendall Morris. Are samples from manipulated sites suitable? Examples would be burned, fertilized, etc.
Definitely. And yeah, that's a great question and something we've thought about, kind of a bit of a little teaser. We are looking at in the future having a call that focuses on sort of post disturbance. And so that more so fits within that post fire scenario. But that is something that is a need within that scientific community to have data, post disturbance, to understand how that affects soil functioning. And we do have current projects that are looking at, fertilization rates, or looking at different kind of more ag focused applications too. So those are all encouraged.
Great. So Sarah, when do people get their data back?
Yes, that's a great question, and one we are currently working on in terms of timelines. Right now, we're in the process of working on a number of scripts and different workflows to speed up the time between when we get the cores and when we can deliver data. But I would say for our more straightforward data types, I had mentioned, the pH, the texture, the ion data, that is largely that we will have that available likely within the first 6 months. It'll get faster as we improve our pipeline, but I would say that because the projects are 12 months, and so that is a bit so we can also make sure we have all the data QC'd and packaged in a way that PIs can take it and then kind of run with it. So be both raw and processed data. I would say at that six month mark, we'll have a lot of the more straightforward data types, and then within the year, we would have all data types available. Maybe the only exception would be our Joint Genome Institute, the metagenome data. We are working with JGI on their timelines to figure out, the right frequency that we're sending them samples, and then they do have their own queue that we have to wait a few months to get data back. So kind of a squishy answer, but I would say if you have an active project, feel free at any time to reach out to me as the project manager. And if we're very open to trying to figure out if you need certain data types for, say, a presentation, or you're a grad student and want to present on some sort of data, we can work with you and try to get certain data types to you earlier if need be. So a bit of flexibility there, but within the larger project, we are kind of in the midst of figuring out those pipelines to get data processed and QC'd more efficiently.
Are there resources available to help people understand the data, the PIs that might submit proposals?
Yes, and that actually ties in really well to The last slide I showed, which is on the MONet Community Science meeting, we realized that some of these data types, a lot of PIs have never seen them before, interacted with them before. It would be kind of wild if someone had used all those data types. There's a lot of different data that is part of that MONet suite. So the community science meeting is really our first step towards providing those resources to the community. Again, the registration is open for remote attendance. We do have some participants who will be on site, kind of get that more hands-on experience, but it is open for remote as well. And that would give people a great idea of how can you utilize these different data types? How can they help answer your research questions, kind of get you thinking about what we're providing and if that is helpful for your program.
So how long does it take to collect soil samples and field measurements?
And I would say that the answer depends, just based off of this first round of projects that have gotten going. Some PIs will go out and have no problems and their soils are the perfect condition to sample. But we've also had instances where there's been a drought in the region the past couple months. And that has made sampling very difficult because the slide hammer and our coring equipment, it is manual. And so if the soil is either super clayey or wet or super dry and compacted, it will take longer to take those cores. And then, as I had mentioned, there's also infiltration rate measurements. And so that involves adding water to a spot. You have the infiltration ring, you add your water. If your soils are, say, super wet and that water isn't going to infiltrate, that will take a bit of time. And we do have some guidance of if it takes longer than an hour to stop the measurement, that you might be there all day if your soil is just not going to let that water infiltrate. So it does vary, and it does depend on how many sample sets you're collecting at one point in time as well. So I think we've had some PIs that are able to kind of go out and sample, let's say within like an hour or two, others where it can take closer to 3 or so. It depends also on how many people you bring out to the field as well to help. But I would say it could take better portion of a day if you have a lot of sample sets and if your soils are not cooperating.
Very good. So Kendallin Morris was hoping that you could reiterate when you anticipate the next call opening.
Yes. So we are planning to have it open on November 6th. We are, as I had said, waiting for a little bit of guidance from DOE. And so we want to make sure we have the most updated information to post, but our plan is November 6th as being that open date. But again, we will let the community know if it ends up being a little bit later. But I would say our team at EMSL is ready, and we are kind of finalizing a few details first. Yeah.
Thank you. Sarah, what if we submit a proposal and then need to change our sampling time? For example, there's changing plans or a major weather event that would prevent access. Can we reschedule?
Yes, and we've had that happen. And so I would say that is very doable, and something that if that does happen, you already have your project in the system. So we'll work with you. We've had a few PIs with this previous call. We'd like them to resubmit just so we have all the proposals for this cycle so we can review all of them and make sure we're not leaving any out. But I would say if we need to reschedule within, sometime during that year, we can work with those PIs and figure something out.
Can you provide some examples of science questions on approved MONet projects?
Definitely. And we have gotten a wide range, which has been really exciting. And so we've had a project that was in Alaska looking at how climate change is affecting the carbon that is within those soils. We've also had another project looking at how different stocking rate of cattle affect the soil carbon that is within those areas. So I would say we've had a diversity of questions, as well as what I showed during the presentation of diversity of soil types and the regions that they're from and the land use that's associated. So it really varies, and MONet is not evaluating the proposals based on the scientific merit. We want the data to be useful to the PIs and their teams. And so that's something to keep in mind as you're considering putting a proposal in, is how does this data help complete the story that you're trying to tell at your sites, and give you more insight into the soil functioning within your research plots. So that is really the top priority, is that you can answer your research questions. And then again, MONet also wants to be utilizing kind of these different sites from around the US and incorporating that into our database and being able to say they've all been processed the same way, they've all been collected the same way. That helps with the comparability. So I would say that the questions range, and we are not evaluating based on that criteria. Really, it is that first come, first serve basis. And then also, thinking hard about your team's ability to take those cores and work with us.
So will the MONet team be at AGU?
Yes, I know John is again on the call, so if he wants to put a plug in for any specific sessions or workshops. Go ahead, John.
Yeah, so we'll be there. You can find us at the EMSL booth that we'll have at AGU. And also there is a MONet related session that I believe is going to be happening on Wednesday, December 13th, that will be focused on data types and big data, MONet style big data. So please join us and swing by the booth, hang out at the poster session, or come to the oral session, and we would love to chat.
Great, thanks, John. Last question actually has to do with this webinar recording, so I can answer that one, and that is, when will it be available? And any EMSL Learn webinar that we hold is available on the EMSL website in the EMSL Learn section of the website, usually about a week after the event. So I would invite you to go and check out that section, and you can also watch prior recordings of other webinars where we talk about EMSL instrumentation and capabilities. And with that wraps up today's webinar. I want to thank Sarah and also remind you to register for the November 15th webinar on terraforms. So look forward to seeing you then. And also last plug for the MONet Community Science Meeting on November 7th and 8th. So thank you for attending. Have a good day.